We're recording the day before Thanksgiving here in the United States. It's bright and early in Eugene, Oregon. It's late afternoon in Italy. I'm in my in law's house in Maine. It's also the first episode where we are recording remotely, so you'll see that there is a sound difference, but we're going to make do.
I mean, I have never made coffee before in my entire life.
That is amazing.
Hi, hello, and welcome to Coffee Literature Review. I'm Chris Hendon, and today I'm joined by Doran Pennington. We have a very special guest today. It's the first time we're doing this. We've invited the lead author, the communicating author, on a recently published paper in the journal Nature Microbiology to discuss their paper with us. The paper is titled Coffee Consumption is Associated with Intestinal Lawson-Bacter Abundance and Prevalence Across Multiple Cohorts. We're really happy to have Professor Nicola Segata join us today from the University of Trento, where he leads a group that studies computational metagenomics. Hi, Nicola. Nice to see you.
Nice to see you as well. Here we are.
Yeah, we're grateful that you could find some time to talk to us today. I was doing a little bit of research, and in my field, Italy is very well known for being one of the leaders in computation, and I think, and also in sort of chemical theory. And I think that recently a pretty sizable investment was made in Verona, the city just south of you, for a new supercomputer, the Leonardo. Do you use this computer for your research?
Not directly. So I'm a computer scientist by training, by the way,
Yeah.
because in metagenomics, studying of the microbiome, 95% of the work is done at the computational level. But no, we have a big high performance computing system in our university, so we use that. Well, big for our needs at least. It's not a
Yeah, sure.
supercomputer.
Well, so another thing that most people probably are unaware of, but happens pretty regularly in Italy, is that many professors go to undergrad at the school, then maybe do a PhD at a school, then maybe a brief postdoc somewhere else, and then come back and become a professor at the school. Are you from Trento?
Yes, I'm from Trento. I guess I'm, however, a bit of an outlier because, yes, I'm from Trento. I did my PhD here, then I went to Harvard, and then that was the point in which in my city there was hiring, and so I came back. But this is difficult to tell also to my trainees because this is not the usual path. I consider myself very lucky, but it's not what we should do. I also need to add that I think I'm lucky that because Trento is in a semi-autonomous region of Italy because it's fully on the mountains, so they have more money, more everything from the central government, and in the past they were able to use them well. So we are kind of considered the Switzerland of Italy. So
I visited Trento before back in maybe it was 2022, just after COVID, and
okay.
It reminds me a lot of like basically these 100,000 people sized towns that you find that have these amazing, I'd say almost medieval like castles dotted around. It's a really nice place. But you also like seem to only go to nice places because objectively Harvard's a nice place.
Of course, yeah. I don't know if you're interested in this story, but basically, so with my PhD was done only fully on computer science, machine learning. Now you need to say artificial intelligence but it's the same thing. And then after that, I wanted to change a little bit and be more applied, solve real problems from biology. And one of the options was going to Harvard, but I didn't know what metagenomics was at the time. But I was lucky enough to arrive when the human microbiome project was really rising, and scientists there were at the point in which they generated the short-term metagenomic data, which means a lot of sequencing from the microbiome. Nobody had an idea about how to look inside that because that kind of data was never been generated before.
Yeah.
So it was the right time because they needed new computational way to look inside that huge amount of data for the time. And so we started developing some new computational tools to make sense of that. And yes, 15 years later, we are still here trying to improve those computational methods, actually. we started at the beginning. We were looking for which phyla were present in our gut microbiome, meaning which big division of bacterial types were there. Now we are at the point in which we are differentiating the Escherichia coli that is in my gut and in your gut by single nucleotides in their genome.
Yeah, it's actually it's amazing.
Yeah, and it's all computational stuff, no? Because the sequencing change in cost dramatically, but the type of sequencing we are using is the same of 15 years ago.
Yeah, sure.
So we've talked previously to an astrophysicist, and they have a similar problem of needing to sift through these enormous data sets to find some kind of signal that they're looking for. And looking through the data in this paper, it seems like it's on an unimaginable scale. You have so many things to look at, trying to find some kind of correlation. And it's a similar challenge. You're just, instead of looking out into space, you're looking into the gut. So I'm curious how you started to apply computer science to this field, and how you came into it from a computer science perspective.
Yes, so there are two main computational steps in this kind of analysis. The first step is going from reads, from sequencing data, to some sort of profiles, knowing which bacteria are there, which genes are there, and so on and so forth. The second step is to basically analyze these profiles, these numbers, these labels with respect to the data of interest, diet, for example. These are two well separated tasks. The second task is a little bit of a traditional bioinformatics, if you like. The data type change a bit, but it can be a bit traditional. The first step, instead, it was something that was not implemented, because basically there was no way to do that. So when I write to Harvard at the time, that was the key thing: developing the first tools, try to make sense of it, and analyzing when they assembled for the first time the human genome. That was a big computational challenge, because you need to basically solve the puzzle of overlapping all the small pieces of DNA to reconstruct the full genome. What we have to do with the metagenome is many times more complex, because we need to solve multiple puzzles at the same time. But at the beginning, we don't know which pieces are coming from which puzzle. No? So the idea is that we need to reconstruct thousands of different genomes from the same samples, in which all the reads are scrambled all together. So that's a little bit the challenge. And another challenge, I would say, is that at the beginning of these, there were only very few reference genomes, so only very few bacteria for which we knew the genome. And so one of the key things in the last years, at least, was to be able to reconstruct genomes directly from metagenomes without the need of isolating the bacteria, and so having references of things that nobody isolated before. And that was another step forward.
Yeah, so for you, is the level of correlation between coffee and the specific species in the microbiome, does that just explode out of the data as being really significant and obvious, or is it something that you really have to go and search for?
Yes, so in general, we are interested in associating features of the microbiome with features of the diet. And so one thing we did was to see how much the microbiome can predict what you eat, one food at a time. This was a way not to use the microbiome to tell you what to eat, because you know what you ate,
Sure.
but it's more to measure the strength of the link. And when we did that, the microbiome and coffee, so coffee was the strongest association with the microbiome. So we decided to go deeper into that. And so the first thing that we did was to correlate then coffee with specific microbes, and that's how Losibacter came out.
But let's go back a step from there, because I think we'll get to the novelty of this paper in just a little bit. The fact that you could I get it down to even like one or two compounds that show up in coffee is pretty remarkable. But let's go back to, so I was doing a bit of research, reading some other papers, and I came across a Nature paper from Peter Turnbaugh, was talking about major dietary shifts could result in rapid changes in the gut microbiome. I'm sure you're aware of the paper. Doran, if you didn't come across it, basically this paper says if you go vegetarian, you see a major fluctuation or change in gut bacteria. And if you go for a full meat diet, then it shifts in a different direction. And my fundamental question is, how quickly does the human respond to different changes in diet?
Yeah, there are many different ways through which diet impacts the gut microbiome on different scales. I think we need to do a step back a little bit, because we need to understand how unique each of our microbiome is. So if you take two persons, even two twins, right, and you ask how many species they have in common in their gut, you're around 20-30%. So identical genome, only 30%, let's say, of the species in common. If you look at the strains of those species, like the variants, and importantly, two strains in the same species may have no more than 50% of genes in common, so very different bacteria. You usually have 0 strains in common in unrelated individuals. So we are at the level in which each of us has a completely unique microbiome compared to the human genome, which is 99.9% identical. However, if I look at my microbiome now and my microbiome in a year from now, almost all the strains will be the same. So our unique microbiome is kept in time at the genetic level. Which microbes are there are not changing too much. However, how abundant each species is can change a lot based on what I ate yesterday, what is my habitual diet, and also if I change the diet. So becoming vegetarian all of a sudden surely will change in the short time the abundance of many species, because there will be species that are depleted because they don't have any meat-based food on which they are growing, but then the microbes are there. So if you change again the diet, that will change accordingly. And then there are other ways through which the food can impact our microbiome, because some foods have microbes on the foods, and those microbes on the foods can colonize our gut potentially. That is a completely different story.
Yeah, sure. But so it seems as though it's adjusting very rapidly.
It's adapting very rapidly the abundance, because every time we go to the toilet, basically we flush out our microbiome. So every time we eat after that, the microbiome is basically reassembling around the new food that we eat. So yes, the microbiome is changing very rapidly, but the microbes that are there are not changing too much in time. I think the key point to highlight here is that not only the microbiome changes a lot between individuals, but it changes a lot in different populations. So
yeah, right.
In microbiome research, it's extremely relevant to whenever you look for the association with the microbiome as something, you need to do it in multiple calls to make sure that what we are seeing is not just a confounding factor or a specificity of one cohort, but is something that is really supported. So in our paper, we put together the UK population, the US population, and also the Italian population. So we can make a lot of fun here about different kinds of coffee we eat that we drink in these 3 different countries. But that was the point. It was reassuring that we see the same exact association, at least with Rosenbacterus sacculiticus, in all these 3 codes.
Yeah. So, but I want to go back to the type of data this is, because like when you're collecting these types of data from this cohort, are people providing you with the information of what they are consuming while this data set is being collected?
Yes, in the predict cohorts, especially, we have very deep dietary data from food frequency questionnaires, but also these individuals are logging on a dedicated app what they eat for several days, and this is all information we have available.
So this sort of thing doesn't happen in chemistry, like this thoughtful construction of data sets that people could use and actually like conclude important things about the world with. Like we just don't do that in our field. So it's hard for me to get my head around that you can get so much data, and we probably could do it in our field too if we actually tried, I guess.
Yes, I have to say that this was powered by a company which is selling personalized nutrition advices and kits. And this is a research project embedded into that, because in the paper we described 34,000 individuals more or less, but we already have availability of almost 200,000 individuals. And this will not be possible with any, usual research grants because of the size of the data.
Of course.
Yeah, I came across this. I just assumed Zoe was some kind of statistical acronym. And then I got to the competing interest section and I saw co-founders of Zoe, and that got me interested and I looked the company up. And so could you, I just went to the page for Zoe and looked up some of the information about it, but could you explain what the purpose of Zoe is, why it exists, and why people might want to be involved with this company?
Yes, so Zoe was co-founded by Tim Spector, which has been working on genetics, but especially recently on nutrition and the microbiome, with the idea that studying the unique biology and microbiology of each of us can explain our response to food. So, the twins that I mentioned before, identical genomes, but if you ask them to eat the same exact food in the morning, and it's actually an experiment that we did on 1,000 twins, more or less, they have a completely different glycemic response. You cannot explain it easily with genetics, maybe a little bit of epigenetics, yes, but as I told you, the microbiome is completely different. So, the microbiome is there also to digest food, process, compounds and stuff like that. So it makes a lot of sense that the link, how you explain the different response, the glycemic response to food, is there in the gut microbiome. Now, I also told you that the human microbiome is unique to each of us, so that's why you really need big numbers to try to find out these links. And so Zoe, what they want to do is to use the microbiome, and not only the microbiome, they have, challenge with the same food, they have a lot of information, and the idea is then recommend you the diet that you want to have for your goals that are specific from you. And if you think about diets, there are a lot of diets that that they work, but only in a subset of individuals. And this makes a lot of sense because, our biology is unique. So there cannot be one diet working for all of us.
So before we get kind of into the outline of the paper, a question that I wanted to ask you is, has your research in the microbiome impacted the way that you think about eating food and the way that you think about your own diet?
Yeah, I would say so. Yes. I am aware about what is the role of the microbiome. I'm aware about the way, the thing that you are feeding not only yourself, but you are feeding your microbiome. Microbiome can be an enhancer of the benefits, but also of the problems of your diet. So on one side, yes. On the other side, we all know about what is an healthy lifestyle. No? So and a lot of recommendations are independent from the microbiome. But yeah, being more aware about the mechanisms and also looking at these experiments in the twins makes you really realize a little bit more why it's important to have a healthy diet, and also appreciating healthy diets and so on. Which doesn't mean that we need to be eat only very healthy diet all the time. That's not the point.
Well, okay, so let's get into this particular paper. It expands upon a previous report from your group, which was used a much smaller data set that showed that coffee might have some impact on the microbiome. And now this paper really nails it down and basically says, independent of whether you're from Europe or America, your microbiome composition depends on whether you consume a little bit of coffee versus a lot of coffee. For me, I want to see if I understand the novelty of this. Is that this really is a good and rare example where you can link a modification of the microbiome in all of the individuals that we looked at, independent of their populations, because we've just heard from you, they're all different. We can link that change to a very specific subset of compounds, molecular compounds that are unique to coffee.
Correct. I think the strength are two. The first one is that what we see is really reproducible across cohorts. The second thing is that is very, very strong. It's particularly strong. It's surprisingly strong. This link between coffee and a specific bacteria. These are the two strengths. Yes.
Yeah, but the bacteria so let's do the bacteria first. So I always have trouble with all the names of bacteria because they just get so long. So I'm just going to call it Lawson Bacter. Is that sufficient for level of fidelity for this?
Sure.
Okay, so
Let's define it at the beginning. Loso for us is Losonibacter asaccharicus. Yes.
okay, perfect. so I'm going to call it Lawsonibacter. Okay, and I was doing a fair bit of reading trying to figure out what this bacteria does. So we know its population is affected by coffee consumption now from this paper of yours, but from what I could glean from this, it produces butyrate. Is that right?
Yeah, there are a lot of bacteria producing butyrate, and butyrate is a very positive thing to have in the gut. So butyrate producing bacteria are usually doing that starting from plant-based diets, and that is very, very important. However, we don't really know much about Losibacter. There is only one official isolate, and this is one of the cases in which someone discovered a new bacterium in the gut. It was not us, by the way.
So I read that paper. I learned that it was isolated from one Japanese woman's poop, just one. Okay.
Yeah.
Now, when I hear that, I'm thinking like, what are the chances, first of all, that we missed it, and why was that Japanese woman provide? That was she in a cohort, and we just happened to find it?
Yeah. So Losingaki is present in 90% of the population on average. So it's not that it's rare. But it's very difficult to cultivate most of the gut bacteria for a number of reasons. First of all, because they are anaerobic, strictly anaerobic most of the time. And in many, many cases, you don't know which substrate they need to grow. So we got interested in Losingaki also because we were able to isolate it ourselves. In the lab by chance, because we were looking for unknown bacteria, and Losingacter popped out. So we also have another isolate of it. So I think the point is that there are still a lot of bacteria that are completely unknown and never cultivated. So it's not surprising that it was isolated only once.
But thinking about whether it's a good thing or a bad thing that you can affect the population of this, one thing you don't see in this paper, and maybe that's by deliberate choice here, is you don't make the statement that it is good or bad that it's been affected by coffee.
Correct. So when we study these topics, there are two steps. Now, so there is the effect of the diet on the microbiome, and then the effect of the microbiome on health. Mm-hmm. And it's difficult to disentangle that, no? Because of course, diet also has a direct effect on health. In any case, we have a ranking of the bacteria based on their association with positive or negative cardiometabolic markers in large populations again. So this is associations, but we have a list of bacteria that are associated with good cardiometabolic markers, so good glycemic responses, low BMI, all these sort of things, and vice versa on the other side. Losingibacter in this ranking comes out basically in the middle. Mm-hmm. Right. So we don't have big evidence that he's a good guy or a bad guy. Doesn't mean that that is really neutral, because it could also be that some strains, some subtypes of Losingibacter are good and some are bad. At the moment, it's not that we try to hide it because of future publication, but we are investigating that. Of course, the other way to investigate it is in vitro. No, so in vitro we are doing a lot of experiments now to try both to understand which are the specific components of coffee impacting the growth of this bacterium, but also what this bacterium produces. Yeah, maybe butyrate, but how much? Does it do it a lot in the gut? Under which conditions? So these are all open questions.
Yeah, I think the one thing we definitely know from this beautiful paper here is that if you drink coffee and you drink a fair bit of it, you're a regular consumer of coffee, that you will definitely grow Losingibacter.
Yes, it's also always about statistics. No? So sure. I'm sure there are some known coffee drinker, but yes, that is actually absolutely the case.
Something I thought was interesting about this specific bacteria is that you make a statement in the paper that around 80% of the never coffee drinking population has Losingibacter in their microbiome. And so I'm thinking, okay, that means we basically all have it, regardless of whether we drink coffee or not. But then you have this other data set of, people that are not in the Western world or people from ancient times. I don't know what ancient exactly means, and almost none of them have this at all. So it seems to be directly linked to drinking coffee, even a few times in your life. So is it the case that if you drink coffee once or twice, you will grow this
That's
bacteria?
a fantastic question. So first of all, the ancient samples are the gut microbiome of mummies or fossilized poo, which are called coprolites. So close to here, there is they found the Iceman is a mummy who is 5.3 thousand years old, and its intestinal content was still available, and we sequenced it. And we found a lot of microbes that are typical of current non-westernized population. But without opening to many other doors here. So your question is great. So, losing bacteria, as far as we know, can live only in the gut of people. Right. So, yes, if you have coffee, you will stimulate the growth of losing bacteria, but you still need to get losing bacteria from somewhere, because when you are before birth in utero, the individual is sterile, so you need to get these microbes somewhere. So the point is that if a population is drinking coffee, probably is boosting the abundance and the prevalence of losing bacteria. But if no one in a population drinks coffee, probably losing bacteria can go down at a certain point in which it will disappear. So if you go into a population who is drinking who is not drinking any coffee, and you drink a lot of coffee, you may not get losing bacteria growing because you are not able to get it from the population. No, it's like pathogens. We have several studies looking at microbiome transmission, and it's like with pathogens. You can get in touch with a pathogen if someone around you is infected. Same thing with the microbiome. You can get your losing bacteria if you are in touch with someone who has losing bacteria in their gut.
So you did some in vitro studies here, right? Because you wanted to parse basically coffee concentration's effect on the growth of this bacterium. The two options you chose were mocha and instant coffee. And I just feel like that's criminal. Given you could have gone and had an espresso and could have achieved something, something pretty strong.
I have a comment. I have a comment on espresso.
Yeah, go on.
So your thresholds for never is coffee intake up to 20 grams a day. And so if you're living in Trento, Italy, and popping to the cafe for a single espresso every day, I think that means that you never drink coffee.
Well, it's the concentration of caffeine. So this is also another fantastic question. No? We had to match the coffee consumption in UK, US, and Italy, and in US you may drink. We had individuals eating liters of coffee. Then you need to understand, how much caffeine is inside that. So it's a very complex matter. And but yes. So I think so. First of all, I didn't do the experiment because I told you I'm
Sure.
a computer scientist. So if I enter in the wet lab, everyone is screaming.
Chaos.
And
Yeah.
exactly. But Roberta Marconi and Viviana Ricci did these studies, and there was a lot of discussion about which coffee to brew. Also, because brewing coffee in the lab is kind of an achievement. It's like you are not allowed usually. You're not allowed to drink coffee in the lab, for safety reason. Anyway, yes, I think we wanted to have a standardized coffee matrix, and so instant coffee and mocha coffee were those that are more easily reproducible.
We have an experimental coffee laboratory, and we spend most of our time thinking about making reproducible coffee. So I don't actually have a fundamental problem at all with mocha versus instant. We make the same decisions. It's not really about consuming this coffee in this particular study. This is about looking at two different strengths.
But you're so much of an expert. We should talk about that, because one problems that we have in the wet lab is that the coffee has particulates inside that, and that is interfering with measurements of how the bacterium is growing because of the particulates of. So we need to filter for doing some experiments. But yeah, probably you should.
Yeah,
get in touch.
That's a really tricky problem, and I hadn't really appreciated that putting particulates in your basically on petri dishes is an issue, but it's
Yeah, well, we have episodes on microplastics and heavy metals in coffee, which you can imagine causing some problems trying to grow bacteria.
Yeah, also because it can be in petri, but we also do in liquid, no? And it could be that some bacteria are growing because they are the particulates, so physically they want to grow. So it's a complex story, and we are working on that to try to improve it. Yeah.
Yeah. Okay. So before we go, I have two more things I want to get at. So we also recorded an episode very recently on the growth of mold on coffee. And this is after it's green. Before it's been roasted. It's green coffee that develops mold, but also brown coffee that's been roasted can develop mold.
Cool.
And it got me thinking when I was reading this paper, it's like people don't want molded coffee in principle because they think it's, something bad has happened to it. But we don't really know what the bacterial population is of that mold. We have no idea, and it could very well be actually a favorable thing. Who knows, right? I have no idea.
Yeah.
It got me thinking. It's like that could definitely affect how your gut responds.
Yes. So also Rosenbacher, we had the question whether it was on coffee, and we are getting Rosenbacher because it's on coffee.
Yeah.
We don't have any evidence that Rosenbacher is actually present in coffee beans or in any step of the coffee.
Yeah. Okay. So that's a good thing to know. Well, then, so the final question really is, from my side at least, is you've done all this work and you mentioned caffeine before. So I guess going in, maybe one of the working hypotheses was caffeine might just be the molecule that's stimulating certain bacteria growing in some way. But then after you read this paper, you very rapidly realize that it's probably not only caffeine that's giving rise to this, but it's a variety of compounds of molecular weight, roughly 174.
Yep.
You were able to isolate, or at least determine some of those compounds that were isomeric and had, or at least had the same weight. And one in particular jumped out, which is quinic acid. Most people in the coffee industry are very familiar with quinic acid, because it's the decomposition product of chlorogenic acid during roast. And chlorogenic acid is unique, really special in coffee. It's like one of the highest sources, I think maybe the only high source of CGA in the world. So finding a decomposition product of a molecule that's specific to coffee is the smoking gun result that caffeine is in all sorts of stuff. It's in tomatoes, it's in bladder, it's in whatever. But if it comes from quinic acid, so then the question for you is, what else has quinic acid in it?
Oh, that's a good question, but I'm not a nutritionist and not a chemist, so I cannot really answer to that.
Well, so the reason I'm wondering it is because you go through in figure 5B, which is, for those that will not read the paper, you can imagine it's all these different compounds and parsed into whether you never drink coffee, you sometimes drink it, or you drink a lot of it. And basically it puts a score on whether these are really important in affecting the Lawson-Bacter population. And you can see caffeine is, of course, important. There are a few other ones that are listed with caffeine that are important, but quinic acid stands alone in its own little subset here, along with trigonelline in another population down below. And it really is these three compounds: caffeine, quinic acid, and trigonelline, and then the other ones we don't know. I kind of know the answer to this, but why don't we know those other compounds?
So this is done with metabolomics in the blood, and so there are a lot of peaks that are not assigned to any known metabolites. I think there are also a lot of metabolites that are not rich in the blood and they are remaining in the gut. I mean, coming back to caffeine, indeed, we don't think it's caffeine. We look at coffee, normal coffee, and decaf coffee. There is some difference there, but of course, decaf and non-decaf drinking individuals have also other, confounding factors, different numbers, and everything. So, but the first results we are seeing in vitro are that probably is not caffeine. We have a confirmation there. Quinic acid is very relevant. So this is what we are trying to do in vitro, because in this figure you're pointing at, the results are clear, but you also see a lot of noise, and these measurements, metagenomics, metabolomics are all quite noisy, and so that's why we need the mechanistic studies to show. Our next challenge will be to try each component of coffee separately, and also each downstream compound to see which one has effect. we know fibers, polyphenols, all these chemical compounds are extremely important for many microbes. We just need to understand which of them are key for losing bacter.
Yeah, and I guess perhaps even key for other ones that you definitely know have positive and negative effects, like E. coli. You know, if you knew that a particular polyphenol would wildly grow a population of E. coli, you might want to avoid it. Perhaps, you know, it's a
Exactly. Yes. Also because this will open the stage to think about prebiotic intervention. Because if you know which compound is stimulating a specific microbe, yes, you can try to use the food with the most abundance of that compound, but you can also think about prebiotic intervention.
So previous episodes, we mentioned a few, but one of the previous episodes that we did, we talked about doing a coffee enema. And this is every time you see a paper on in the peer-reviewed literature, it's basically because someone put hot coffee inside their colon and it hurts them and they end up in the hospital, or worse, they can end up dead. I was thinking about this. It was like, you know, usually it goes through your mouth and it goes through the pretty acidic environment of your stomach and then eventually ends up in the small intestine. And that's really where you're seeing these populations of bacteria. What do you think happens if you just skip the mouth and the stomach and you just go straight with coffee straight into the rectum?
That's a question we had, because when we did the experiments in vitro, the first question was how much coffee should be put at. So what is the concentration of coffee arriving to the colon? And we are not as expert as you, but we look into the papers and we tried some concentrations. Of course, in vitro everything is artificial. So, but that is a fantastic question. What's the amount of which of these compounds, is the concentration of coffee arriving to the colon and to different parts of the colon, because our gastrointestinal tract is colonized by different bacteria in different locations. So also all our studies are done with the stool sample, so which is a proxy of the whole gastrointestinal tract. So also going in the specific locations will definitely be more insightful.
But so one of the things that you've also been part of is the impact of fecal transplants, which is another thing that was kind of popular. Maybe I'd say it was in the mainstream fad media about the time you got to Harvard, right? It was about 2010. People were really talking about this idea of fecal transplants, and you wrote a series of papers on that topic, or at least one that I remember. Uh, and basically, that's what. Can you tell us what a fecal transplant is and what the outcome was there?
Yeah, so the fecal transplant is what actually the two words says, unfortunately. So is getting some stool from unhealthy individuals and put into the colon of usually unhealthy individual. This is clinical practice for some diseases. So for infections, so for example, for Coccidioides difficile that are resistant to antibiotics, the fecal transplantation is what is actually saving a lot of life. Is really very effective and is clinical practice in many different hospitals. One extremely relevant aspect of the microbiome is that you can modify your microbiome. You cannot modify your genome, generally speaking, but you can modify your microbiome. Now, knowing how to modify your microbiome is way more difficult. Diet can be used to change your microbiome. Probiotics can potentially use to change your microbiome, but we still have to learn very precisely how to do that. Fecal transplant is what can change most, impactfully your gut microbiome. And so in some diseases it's working well. One of the condition which fecal transplantation is extremely promising is something very advanced, which is improving the success of immunotherapy against cancer. So cancers like melanoma, advanced melanoma, have been revolutionized, usually the treatment with immunotherapy. That works very, very well in some individuals, but unfortunately not in all of them. What it has been shown is that the microbiome has an impact on whether one will respond or not to immunotherapy, which makes sense, broadly speaking, because immunotherapy is acting on the immune system, and the immune system and the gut microbiome are two faces of the same coin, if you like. And because of this, there are clinical trials trying to do fecal transplantation into individuals that are not responding to immunotherapy to see whether they will start responding. And the first evidence is that a good fraction of these non-responders become responders. And this is huge. It's changing the way we are thinking about treatment for advanced metastatic melanoma or other cancers. So
this idea that you can have these cells living inside your GI tract, and these cells can dictate your human health, and you can control these cells, but you can't change you. You can't change you, but you can change them. And if you change them, they can change you. And you've got a paper here saying just if you drink coffee alone, you can change it. This is a big deal. This is like the future of medicine, basically, not for all diseases, of course, because not every disease is going to respond the same way, right? But who knows?
yeah, absolutely. It's also philosophical if you put it that way, because there are many diseases that are called non-communicable diseases, like diabetes
Sure.
or condition like obesity. And some of these conditions are linked, at least partially, with the microbiome, and maybe causally in some cases. And we've shown in other papers that the microbiome is transmissible. If you start living with someone in the same house, after a few years, you are sharing 20% of your microbiome from starting 0% of your microbiome. So, and this was brought forward also by others, some non-communicable diseases may actually be partially communicable because of their link to the microbiome and because the microbiome is transmissible. So yes, our microbiome is modulated by transmission, by diet, by everything. If we arrive to the point in which we know how to change the microbiome reproducibly, we can really have a huge impact into risk for diseases themselves, and even treatment.